Great. Hi, I'm Christian. I'm working with Katrin and Timo in Jülich. And I'm working on the development of, mostly working on the development of AI methods for automated analysis or parcellation of cytoarchitecture. I talked about the methods that I developed in previous meetings. This one will be a little bit different in that I want to basically take a kind of a break, look at what we did so far, what is there, and what I think might be good future research direction. And this is also something that I would be very happy to discuss if you think that the things that I'm showing make no sense at all. So feel free to point that out. So let's start with the data that we're working with. So this here is a cell body stain section from BigBrain, scanned at 1 micrometer resolution. And what we are interested in is the cytoarchitecture. I'm mostly working in the cortex, but most of the methods can also be transferred to subcortical regions. So when we pick out a part of the cortex at this very nice border between V1 and V2, which is the only border that even a computer scientist can show without getting into trouble, we can see that there is a more or less prominent transition in the organization of the cellular architecture here. So we can define borders, and if we continue doing this for more brain areas, we end up with many borders and potentially at some point with a parcellation. Why do we need this parcellation? Most of you will be familiar with this. These parcellations are the basis for building brain atlases, as this one here, the Jülich Brain Atlas. So parcellations created on the individual brain sections go into this kind of atlas. And here it's important that since we have a quite large inter-individual variability, the more data we can feed into these atlases, the better. So of course our N will always be comparably low compared to in vivo studies, but still we should be able to analyze the post-mortem data that we have available. And as it's often the case, this annotation procedure is quite time intensive. It requires expert expertise. This is why I come into play in trying to automate this step of identifying or characterizing the cytoarchitecture. So the basic idea is quite easy. We take image patches from our cell body stained images. We apply deep neural networks because the cytoarchitecture is quite difficult for a computer to actually describe precisely enough to do it with classical methods. So we resort to using deep neural networks which do this step for us, and then we end up with a segmentation or classification of some kind. Of course, if it was that easy, I wouldn't be giving this talk, or I would now show you a fancy complete brain atlas, but it's actually not so easy. So we tried a lot of methods, starting with a very simple one where we just tried to classify or segment, and then went through things like self-supervised learning and more sophisticated things. I unfortunately don't have the time to show you all of these here, but the summary is that the most important one, brain mapping is very challenging. The reasons for this are multiple. The patterns that we need to analyze to characterize cytoarchitecture are complex and have multiple scales. We have processing and inter-individual variability that need to be handled, and as it's often the case, the annotations that we would need are limited and also partial, and on top of that, we are dealing with very large data sets of thousands of sections in the range of terabyte and even petabyte. How can we deal with these challenges? So the last one is the most straightforward, more or less. We use supercomputers which introduce almost as many challenges as they solve, but without them, it's just not possible to do this task. So this is for the big data aspect. Now, for the other ones, let me introduce two of the methods that we developed. One of them is this one here, which is based on so-called based on the idea of so-called local segmentation models. Here, we started with the premise, okay, trying to identify brain areas in many brains, many different brain areas is challenging. So let's do something simpler. We broke down the task into a set of easier tasks, smaller tasks, easier tasks, and we did this by saying, okay, instead of trying to identify many brain areas and many brains, we try to identify just a single brain area in a spatially restricted part of the brain. In our case, these are these blue blue lines that you see at the bottom, so sparsely annotated brain sections. And then we use each pair of adjacent annotated sections to train so-called local segmentation models, which are deep neural networks, which are basically designed to overfit on the specific architecture in this region, and thereby are able, hopefully, to very precisely classify or segment the sections in between. You can basically think of them as speeding up the mapping process. This actually works quite well. This method was the basis for some of the maps that you can see in the eBrains viewer, and there are also at least 2 posters here in the poster session later by Alexei and Olga, where you can see how this method can also be used to generate maps in subcortical regions. Another method we started with going back and thinking about, okay, what are we asking our neural networks to do? We give them patches like this and ask them to classify a brain area, which even from a human perspective, from an expert perspective, is quite unfair, because we have no idea where this patch comes from. This information is just not available to the network, which actually makes it surprising that they work as well as they do. We try to overcome this by reformulating the problem, in that we said, okay, we have a brain, we can create a surface. This here is BigBrain, but we can come up with a more or less accurate surface for any brain that we have. And this brain is a mesh, and for this mesh, we know where each node of the mesh, or which location in the original histological sections each point in the mesh corresponds to. Meaning that if we have a feature extractor, a neural network that can give us a description of cytoarchitecture, we can basically compute this in image domain and assign it to the mesh, which gives us some kind of feature. And since this is a mesh, we can interpret it as a graph, and in our case apply graph neural networks, graph transformers, or anything like this to basically come from this patch level to a more global level and integrate more information. So these are 2 methods that we developed. Now let's have a look a little bit into what we plan to do in the future, or what I think would be good to do in the future. For this, let's conceptualize again what we are doing. So we start with images, we give them to a neural network, and we get a postulation, which is fine if we only want to do a postulation. Now, what I think would be good to do in the future is to start with images, pass them through a neural network, obtain a feature representation telling us something about cytoarchitecture. So this actually shifts it a little bit in that we do not directly go to a postulation, but we first stop at an intermediate representation which tells us something about cytoarchitecture. From there, we can still go to a postulation if we want. So we are not losing anything. We are only getting a more controlled intermediate step that can hopefully help us to achieve something. So we can think of this as going a more data-driven approach of approaching this idea of characterizing the cytoarchitecture of the human brain. And they actually do not replace postulations, they in some kind just complement them. And actually one interesting thing that I found while researching for writing up my PhD thesis is that von Economo in his work from 1925 has a whole section where he describes an ideal brain map, which he basically says imagines as a stack of paper, each of them having a very specific property of cytoarchitecture drawn onto, which is of course not possible in 1925, but nowadays this is just a feature vector. We just need to find the way to get this feature vector. So how can we learn the features? We need an objective. In any case, whenever we are doing deep learning, we need an objective. Even if we are not doing supervised training, we need some way to tell the neural network what we want from it. We want to learn cytoarchitecture, but this is not differentiable, so to say. So there are multiple ways to do this, and we did this partially, so I'm moving now from what we did, what we are doing, to what we might do. We can use labels, which is fine because it's very close to what we know about cytoarchitecture, but it's in the name, it requires labels. We can use probabilities. We can use the probabilistic maps from the Jülich brain atlas, as they already give us quite a lot of information about which areas are similar, which are close to each other, so we can exploit this with the right formulation, for example using contrastive learning, to build an objective function. One thing that we are currently doing is trying to go one step further and learn from space. So we say locations that are close to each other are very likely cytoarchitectonically similar. So maybe we can exploit this to define a training objective. We can even go a little bit further, and this is now going into potential things that we might do. Learning from multimodal data. So if we have multiple modalities, probably mostly microstructural, for example, connectivity-based data, we could use this to define appropriate learning objectives to build interesting feature space. And the last thing, which I put a question mark, is learning from text. So a few years ago, I would have said, okay, why text? We are dealing with images, why text? And actually, Katrin walked into my office a month ago and asked, can we do something with text? These large language models are very powerful. Can we use this somehow? And actually, it might be possible because there is a large amount of literature describing cytoarchitecture, describing brain architecture. If we can find the right way to pair this, there are models like CLIP, GPT-4, things like this, that can work with text and images. If we could leverage this somehow, this would be a very powerful means, not necessarily to learn the features, but potentially to explain them to us. Because we can learn features, but we don't know what they mean. But if we have like a model, like a GPT model that we can attach to it and just ask what are these features, this would be very, very great. So I'm sorry for going over time a little bit. But with that, I'm already at the end of my talk. Thank you very much for your attention.
Thank you so much. Lots of very interesting stuff. Do we have any quick questions? If not, then I think we'll go. Oh, Gab, go ahead.
Sorry. Oh, just a quick comment. Very nice presentation, Christian. Last element that you brought up, text. There are text parsing softwares, so they do exactly what you're saying, like in the Fraunhofer Gesellschaft, Martina Pitius-Hoffmann, actually reading abstracts over thousands of papers and putting the information in a knowledge graph with independent modalities. You don't have to start from zero.
Exactly, something like this would be the basis for this. I was more thinking about figures and captions, because you would actually have like a pair of, this is the image and this is how people describe it. And if you combine this with a very powerful pre-trained image model and a pre-trained language model, somehow pair these somehow and then just ask it, what does this feature tell me? So this, but with the, I'm very excited to try this, but it's like a whole new domain.
Good luck. Thank you very much.
Thank you.